while I'm introducing you. So Shail, I'm very happy to introduce him. He is a research engineer in neuroimaging with a focus on MRI and PET imaging in humans to understand how many systems, how memory systems work and interact in healthy and pathological populations. So he has an engineering degree from Mauritius, and after his PhD from the University of Manchester in Imaging Sciences, he joined actually my research team in 2012 with the aim of exploring imaging biomarkers in patients with Korsakoff syndrome and alcohol use disorder. So he has taken a particular interest in exploring how alterations of thalamic nuclei play a role in disrupting PAPES circuit or the extended hippocampal circuit. So he's going to speak today about thalamic nuclei quantification in Korsakoff syndrome, trends, pitfalls, and caveats. So Shail, whenever you want.
Okay.
Perfect, go ahead. Thank you.
Okay, hi, good morning, good afternoon, good evening, from wherever you are in the world. It gives me great pleasure to be here for this conference. It's been an amazing symposium so far. Thank you for everyone who has participated, and to the presenters, moderators, and everyone else. I'll be talking to you about my experience with quantifying thalamic nuclei in Korsakoff syndromes. I'll be talking to you about the struggles I've had and some exciting results I've had as well. So without further ado, what I can tell you is that to start with, the Korsakoff syndrome is considered as a complication from alcohol use disorder. The latter itself is a consequence of chronic and excessive alcohol consumption, where patients develop impaired cognitive abilities that includes episodic memory, executive functions, and working memory. And what differentiates uncomplicated AUD patients from Korsakoff patients is that they have added thiamine deficiency, potentially with bouts of Wernicke's encephalopathy. And their main cognitive difference is that their amnesia is irreversible, while AUD patients typically recover with abstinence. Two brain circuits are principally involved in both clinical forms, which are the frontocerebellar circuit, which subtends working memory and executive functions, while the PAPES circuit subtends episodic memory. The thalamus, as you can see, which we know is a relay organ consisting of several nuclei, is found in the middle and shared among both these circuits. And one would expect differential alterations to thalamic nuclei that would drive the specificity of the amnesia observed in Korsakoff patients. And the first insights of the involvement of thalamic nuclei playing a key role in amnesia of KS patients were first through postmortem studies. Indeed, as early as the 1950s, a study involving 70 patients with Korsakoff syndrome, where 90 of them had a history of alcohol use disorder, had lesions in the mediodorsal nuclei and the mammillary bodies. In fact, the mammillary bodies have been systematically present in most studies. And it even supported an initial belief that this would be the main hallmark of Korsakoff patients. Above the mediodorsal nuclei, there were also lesions observed in the pulvinar and in the lateral dorsal nuclei. And surprisingly, none of these studies had yet mentioned any lesions to the anterior thalamic nuclei. However, the early 80s saw case studies in two very well-described Korsakoff patients, first reporting lesions to the medial thalamic group, which was believed to actually contain the anterior thalamic nuclei. And as neuropathological methods became more precise and patients were being more carefully selected, there was increasing evidence towards lesions of the anterior thalamic nuclei being observed. In fact, the last very comprehensive study by Harding back in 2000 systematically analyzed neuronal counts in AUD patients without any bouts of Wernicke's encephalopathy and Korsakoff. as well as patients with Wernicke's encephalopathy and patients with Wernicke's encephalopathy and Korsakoff syndrome. And they were the ones who clearly showed that the anterior thalamic group was specifically affected in patients with Wernicke-Korsakoff syndrome, as compared to damage to the immediate dorsal nuclei and the mammillary bodies, which remain comparable between these groups. So globally, the history of the literature of postmortem studies is quite disparate. At the same time, it is understandable as they span a few decades where evaluation techniques, both in terms of measurements and cohort selection, improved over time. Importantly, sample sizes remain small, and such experiments would give information on the end story, but not how actually these pathophysiological mechanisms would unfold over time. And this encouraged neuroimaging studies in vivo. The first study used the CT scan and showed that the density of the thalamus in 7 patients with Korsakoff syndrome was significantly less than in AUD patients. And this decrease in density actually correlated with memory performances. Sullivan and Perferbaum used MRI at 1.5 Tesla to show that the volume of the thalamus in 5 patients with Wernicke-Korsakoff syndrome was less than those in 24 UD patients. And one MRI study carried at 3 Tesla was the first to use a voxel-based profile to show gray matter alterations in 11 TeS patients that likely came from a mixture of both the anterior thalamic nuclei and the medial dorsal nuclei. So to recap what we know so far, and what we've been able to gather is that the thalamus plays a key role in amnesia, and that it is shared between the frontocerebellar and the Papez circuit, and that finding the specific nuclei, which belongs to either one or the other circuit, will help us unravel the pathophysiological mechanisms specific to amnesia. But it's been over 130 years now since TeS was first described and reported, and neuroimaging techniques have spanned over 40 years, and it's been relatively hard to locate and quantify thalamic lesions at nuclear level. Why is it so hard? Well, a typical image that you see most of the time of the thalamus looks like this, but this is a very simplified version. A more realistic one would be something like this, where you have something like 50 nuclei packed in an ovoid structure that spans cross-sectional areas that doesn't exceed 20 to 30 millimeters in each perspective view. As a result of that, relaxation times and protein densities on MRI measurements are very close to each other. The nuclei appear blurred, offer poor contrast, and even poor signal-to-noise ratio. And what is also important to consider is the pragmatic point of view. The current situation in clinical research is that few studies start or have started with alterations to the thalamus as the main region of interest. Other sequences would be desired to have the full picture of mechanisms happening within a pathology. For example, one would be interested in whole brain volumetric measurements, would even be interested in measures of white matter microstructural integrity, or even cerebral blood flow. We would therefore need many sequences and would potentially have to sacrifice a high resolution, which would require high acquisition times. And when considering a lower resolution image, one would need high expertise in thalamic cytoarchitecture to be able to distinguish between thalamic nuclei, over and above the fact that these processes is time-consuming and prone to human errors. Automatic segmentation methods would therefore be preferred, which would use particular properties of an image, like protein densities, jitter volumes, and even structural or functional connectivity measures. Well, before I look at some data where we had some fun measuring thalamic nuclei volumes using different atlases, I will present to you a method we used using DTI to actually segment the thalamus. The method has been around since 2003, and it is quite smart in the sense that it does not really rely on contrast between nuclei, but evaluates the number of connections between the thalamus, which is defined as a seed region, and any reasonable number of targets. And to run tractography between the thalamus and those targets, and based on which thalamic pulses had the most connections to a target, the thalamic pulse is considered as being principally connected to that target. With this in mind, we used principal brain regions that form the frontocerebellar Papez circuit on 20 controls, 26 AUD patients and 16 patients with Korsakoff syndrome. We ran probabilistic tractography on those 5 targets, known to belong to the frontocerebellar Papez circuit. And we saw that for the frontal executive target, it was principally connected to a parcel that contains 70% of the mediodorsal nuclei, whilst the thalamic parcel that connected mainly to the hippocampus housed 84% of the anterior thalamic nuclei. And we validated these connections via running tractography again to show that the white matter tracks connected does actually make sense. And when we compared connectivity and volumetric measures of these thalamic pulses, Ancover showed that what was principally the mediodorsal nuclei was atrophied in both AUD and KS groups, and to the same extent. The anterior thalamic nuclei, on the other hand, had greater defect in terms of connections from controls to AUD to KS patients. But atrophy was found to be significant in KS patients only. Now, before I discuss the implications of those results, let's see the data that I told you is actually just fresh out of the oven. It was an effort thanks to the Thalamic Segmentation Group, who have inspired Dr. Anne-Lise, Pitel, and myself to look at the precision of atlas-based methods for volumetric measurements. The rationale for doing this was to see how the choice of an atlas would drive the results. While it is true that we didn't have a gold standard via manual segmentation, we would actually like to have a professional like Tomatodias who would actually be able to give us great thalamic nuclei parcellations. We do have history of diencephalic amnesia on our sites and the hallmarks of the regions involved. So in free TMRI data, we compared volumes of the anterior thalamic nuclei and the mediodorsal nuclei, which we know are principally affected in alcohol use disorder. And we also used the LGN and the MGN, which are not normally affected, and to compare and contrast the differences in methods across populations. And these nuclei are all available as parcellated regions in all those 3 atlases, which were the Morel Atlas, the Atlas in Free Surfer, and Thomas Atlas. What we saw was that for the anterior thalamic nuclei, Morel and Thomas methods showed the expected difference between AUD and KS. And none of the methods filled the hypothesis for the mediodorsal nuclei volume. And Free Surfer was the only one who highlighted the difference between AUD and KS for the LGN volume. When we had a closer look at the data, tried to understand why we were getting these results, what we saw was, well, these are 2 MRIs. On the left is healthy control, and on the right is a Korsakoff patients. You can already appreciate the enlarged ventricles and the small thalamus that accompanies this patient. The anterior thalamic nuclei was the only one that had a good overlap amongst the 3 methods. So the Morel Atlas in blue, Thomas in green, and Free Surfer in red. And predictably, since they overlap well, those methods did not differ in volumetric measurements. When in the control groups, they gave more or less the same results. However, when we went to the pathological population, Free Surfer seemed to overestimate the anterior thalamic volume. you might see a slight misregistration, but this is just my visual, which I've made wrong. It's on the native space, it's just due to the image format that this happened, but on testing, it actually is well registered. The reason why this this increase in volume was present in free surfer was probably because they don't really segment the gray matter from the white matter within the thalamus, and the thalamus actually contains almost, in some cases, 50% gray matter and 50% white matter. So whether these white matter voxels are actually included and make a difference, that's probably one of the reasons. And there's also nevertheless the problem with registration, which is known to be difficult in deep structures. And there's a special algorithm for actually running the deep structures for the thalamic segmentation. So we should probably be careful about all this when we are looking at when we are choosing the method for analyzing our data. So now back to the connectivity and volumetric measurements. So what can we say about the results we found? Basically, the graded disconnection and the atrophy, which is similar in the mediodorsal nuclei, but only present in the anterior nuclei. So our study showed that the chronic and excessive consumption of alcohol caused moderate to mild atrophy in both the mediodorsal nuclei and the anterior nuclei, which in turn explain the mild to moderate deficit seen in terms of both working memory and executive functions, and also episodic memory deficits. And this is likely due to the direct neurotoxicity of ethanol. However, patients had further severe atrophy in the anterior thalamic nuclei. And if we take that together with thiamine deficiency or altered thiamine metabolism, this could be why that the mediodorsal nuclei alone wouldn't be responsible for amnesia, but also the involvement of the anterior thalamic nuclei, which would be affected by both thiamine deficiency and alcohol consumption. We know from animal models that anterior thalamic nuclei disruptions, which has been anyways replicated in neuropathological studies, is related to inflammatory processes within the thalamus of animal models of Korsakoff syndrome. And whether this actually could be another reason besides volume changes or disconnections, and contribute towards amnesia. This is probably where future research is going. So a few important points to conclude. Animal models have shown that ethanol toxicity alone is not enough for amnesia. We have seen that the anterior thalamic nuclei is affected in Korsakoff syndrome in a pathophysiological process that starts with a disconnection and then cascades into atrophy. Other processes within the thalamus may be present. Among others are neuroinflammatory processes or alterations to cholinergic efflux that is known to be related to the observed thiamine deficiency. Future research should therefore explore the contributions of these processes towards alterations to thalamic nuclear groups and how they disrupt the papillae circuit in general. How can this be done? First, at thalamic nuclear level through carefully selected cohorts. And next is via a more integrative approach, where we are not only looking at the thalamus as an entity, but by analyzing the brain circuit that it is related to, and also not limiting ourselves to structural imaging modalities, but also molecular ones like PET imaging. With this, I would like to end my presentation by thanking you all for your attention, and also the works that are presented to you is the result of a wonderful team that I am part of. And I cannot thank enough Dr. Anne-Lise Pitel for her support and enthusiasm. And I'm also taking this opportunity to thank our mentors from Stanford and my dear co-founders of this amazing Thalamic group, Manoj and Julia. So thank you again for your attention, and I'll take questions. Thank you.